Hello and welcome. My name is Patrick. I will be your presenter for today. I am going to be talking to everyone about coronavirus 2019, or Miss Rona if you're nasty. So disclaimer, I am not a virologist. I am not a doctor or any kind of medical professional. I am not an epidemiologist. I am merely a nerd who had a lot of time on his hands and decided to do a bunch of research about this stuff. Also note that a lot of this stuff is constantly changing. The research is always updating, and it's coming so fast that a lot of this stuff could be out of date by tomorrow. So, just keep that in mind as we go forward. But let's begin. What is severe acute respiratory syndrome coronavirus 2? Well, SARS-CoV-2 is the virus that's caused the pandemic of coronavirus disease 2019. So that's what we're seeing all around the world. That's what you hear on the news, literally everywhere. It belongs to a family of coronaviruses, which are the cause of a number of infectious diseases among birds and mammals. And coronaviruses are responsible for some human diseases, like the common cold, which are rhinoviruses, as well as more lethal ones like SARS-CoV and MERS-CoV. Like all coronaviruses, SARS-CoV-2 is enveloped, and it has a positive sense single-stranded RNA genome. Note that viruses cannot replicate without hijacking the molecular machinery inside host cells. Thus, their goal is to infect a cell and take over its functions. So a virus is not alive like you or I. In fact, they're not alive like a lot of bacteria, which can have their own replication machinery inside them. They need to go inside a host cell and take over that machinery so that they can replicate themselves. So this is a very close-up look of what severe acute respiratory syndrome coronavirus 2 looks like. So you can see on the left here, we have an electron microgram shown in very pretty colors that you can clearly see the spikes around the virus itself. And on the right is an artist rendition of what the virus would look like. The big orange things are the spike proteins, which we're going to get into, and I'm going to explain exactly what that is in a bit. Essentially, the reason it's called coronavirus is because it looks like those spikes kind of look like the spikes on a crown. So it's like, yes, queen. She's a queen. Corona queen. Anyway, this is a bit more of a diagram showing what the SARS-CoV-2 looks like. So again, those are the spike proteins. There are some other membrane-bound proteins, and the RNA genome that is also covered by some other proteins to protect it. And then, of course, the envelope, which is made out of phospholipids, just like our cellular membrane has a phospholipid bilayer. This virus has one as well, and that's where it contains the genome. So whereas our genome is double-stranded DNA, their genome is just one single strand of RNA that just loops around and is covered by those proteins. So where did the virus come from? And we have this cute little picture right here of where we think it might have come from. So the first human to contract the illness was likely in the Huan seafood wholesale market in China's Wuhan province, where a number of live animals are sold. Now, like with SARS-CoV and MERS-CoV, bats are thought to be the likely reservoir species, the natural reservoir species for SARS-CoV-2, and were likely transmitted to humans through an intermediate host. And then there was subsequent spillover infection. So the reason we think there's an intermediate host is because we haven't been able to find a strain of the virus that has enough similarity in its spike protein, which is essential for it to actually infect cells. We haven't found any strain of the virus in bats themselves that have enough of a sequence similarity in that for us to be confident that that's where it originated from, or like before it came into humans. So it's likely that there was a spillover, that there was a intermediate host, which is very common. That's what happened with SARS-CoV. SARS-CoV, their intermediate host was identified as the civet, which is kind of a really cute little jungle cat. And MERS-CoV, it was camels that were the intermediate host. So we're trying to figure out what the intermediate host for SARS-CoV-2 is. Pangolins have been proposed as a possible intermediate host after researchers discovered the coronavirus with the spike glycoprotein. So again, the spike is the thing that does the infection, contained a receptor binding domain that is 99% identical to the one in SARS-CoV-2. The researchers that discovered this were a little shady in their actual presentation of this. They actually originally said, we found a virus in pangolins that's 99% identical to SARS-CoV-2. No, they didn't. It was only this particular section of the spike protein that was 99% identical. So, it's still not positive if the pangolin is actually the intermediate species. Now, researchers showed that the coronavirus found in the species called Rhinolophus affinis, which is a type of bat. And I have a cute little picture of it here on the side. It has a real fucked up face. It looks like it sneezed too hard or something. It has a 96% sequence identity to SARS-CoV-2. So that is much, much higher than any of the sequences that we've identified of coronaviruses inside pangolins, the highest of which has a 92% sequence identity. So that means that pangolins, it might not be likely that they are the intermediate species after all. What is the intermediate species? We're still not sure. There was another study that suggested that maybe snakes were the intermediate species. That study has been debunked, so that is unlikely as well. Now, how does the virus actually infect a host? So this is going to get a little nitty-gritty in the science. I will try and explain it as best I can. So essentially, SARS-CoV-2 infects cells that express the ACE2 protein. So a cellular membrane-bound enzyme expressed especially in type 2 alveolar cells of the lungs. So what that means is the cells that line the inside of your lungs, especially the alveoli, the little sacs that pick up the air, the oxygen, when they expand, they take in oxygen. Very, very important for you to be able to breathe properly, as you can imagine. The very layer on top of those cells that makes up those cells express this protein. So this protein actually sticks out a bit and goes through the membrane of those cells. And that protein is ACE2. All right, so SARS-CoV-2 needs to attach itself to ACE2 in order to infect a host cell. Now, it's the viral spike glycoprotein that has the high affinity to ACE2, and that is what is going to be doing the binding. So, like I was mentioning, it's very important. The spike glycoprotein is what's doing it. So why do I switch between glycoprotein and protein? The difference in academically there, it's important to differentiate, but like in common parlance, it's not that big a deal. But basically a glycoprotein is just a protein that has had some carbohydrates added to it. So like some sugars that have been added to it. And that's a very normal occurrence with a lot of proteins that are found on membranes. Anyway, that's the only reason I mentioned that. So these cells that are in the alveoli, they have on their cell membranes an abundance of this protein. So it is a cell membrane protein. So it's the membrane that actually delineates the cell from the outside. All right. In that membrane, there is the ACE protein. Okay. So it's very important for SARS-CoV-2 to be able to recognize that protein because that's what it's going to be binding to. That's actually how it's going to be infecting cells. So the way it does that is through the viral spike glycoprotein that I've been mentioning a lot now. It has a high affinity to ACE2 and will thus bind to it. Now, another enzyme that is very important is the cellular membrane protease TMPRSS2. Primes the spike by cleaving it, exposing the fusion peptide. This is actually a diagram right here. So yeah, this is the spike protein right here. This is the segment that actually binds to ACE2. So once it binds to that, then TMPRSS2 is going to actually cut the protein in two. And then that opening of the protein is going to expose another part of the protein that actually is the fusion peptide. So this is the peptide that's responsible for actually fusing the membrane of the virus with the membrane of the cell. All right. I also wrote in parentheses here, and possibly furin. That's another protease, so another enzyme that cleaves proteins that has been shown to be very, very effective at allowing certain viruses to enter our cells. Now, basically, there are some researchers that showed that SARS-CoV-2 actually has a sequence within its spike protein that is shown to be a possible cleavage site for furin. And if it's also cleaved by furin, then that would mean that it is much better at infecting cells than SARS-CoV, the SARS from 2002, 2003, 2004, because that strain of the virus does not have this actual motif. So that's just one of the possible explanations as to why we're seeing such a larger pandemic with SARS-CoV-2 versus just SARS-CoV. All right. Now, at this point, the viral envelope fuses with the cellular membrane because the fusion peptide has been exposed and allows the viral RNA to enter the cell. Now, new research suggests that spike might also bind to cell membrane protein basogen to enter host cells. This one is kind of new that they've discovered. I don't know much about it either. It's possible that it's just kind of as a mediator, so it's another protein that's involved in the whole binding, recognizing, and fusion of the virus to be able to get into the cell. Or it could be something that it can do instead. I'm not really sure, and researchers aren't really sure yet either, so there's going to be more coming from that. All right, so someone just asked what furin is. So again, furin is just another cellular protein, so another protein that our cells produce naturally. And it's produced especially quite a lot in the lungs. So, and there are some viruses out there that use furin to cut them open and allow their genetic information to enter the cell, just like how SARS-CoV-2 uses TMPRSS2 to do it. We think it might also use furin, which is another cellular protein, to do this as well. All right, so now it's in the cell. The RNA is in the cell. Now what is it going to do? It's going to start replicating itself. It doesn't give a shit about the normal cellular processes. That's not its business. Now it's going to take over, and the cell is going to be all about the virus. So I have a very nice diagram here. This is a diagram that is, it's shown for basically all coronaviruses, how they work. We don't have a detailed like kinetics of how SARS-CoV-2 works in this way yet, but because it's pretty highly conserved amongst different coronaviruses, it's very likely that it's like this, or at least very similar to this. So anyways, it is a positive sense single-stranded RNA virus. So what does that mean? Well, basically, DNA is double-stranded. We have two strands of DNA that go around each other, loop around each other, but RNA is single-stranded. And when it's single-stranded, it can either be positive sense or negative sense. Positive sense means that it can be used right away by our cells machinery to actually make proteins. Because the way that we make proteins and we make our whole bodies, essentially, is we have DNA. DNA gets translated into a single-stranded RNA, and that RNA gets, the DNA gets transcribed into a single-stranded RNA, and that RNA gets translated into a protein. That is how all of our proteins are made. So because it is a positive sense RNA, that means it can be translated right away. Whereas if it were a negative stranded, single-stranded RNA virus, it would have to make positive strands of RNA first so that they can be translated. It might be a little complicated. The mechanics of it aren't super important. The big thing that you need to know is that it's just a positive single-stranded RNA virus, all right? And that it's ready, and that its genome is ready to make viral proteins right away, okay? Now, one of the other things that is important for these RNA viruses is that its RNA genome has a very high affinity for our cell's translation machinery. So all of the proteins, the ribosomes, everything that come together to make our proteins, all right, they actually have a much higher affinity for viruses than they do for our own messenger RNA, our own RNA that we make to make our own proteins. So that's part of the reasons viruses are so successful and so deadly. So it will produce virulence factors, which are a kind of protein I'm going to explain a bit more later, and some structural proteins, and most importantly, the RNA-dependent RNA polymerase, RDRP. So if we look at the diagram, we see here we have the coronavirus. It is going to bind to the cellular receptor, and then it's going to fuse its membrane together and release its genome. All right, so the positive strand, the positive sense RNA. And then that is going to be translated by the ribosome right away to make proteins. And one of the first proteins it makes is the virus polymerase, so the RDRP. And that is then going to make negative sense RNA. So for a time, you're actually going to get double-stranded RNA. You're going to get, because the way that transcription works, the way that any polymerase, RNA or DNA polymerase works, is that you can only go from one sense to the other. So you can't take a positive sense RNA and just make more positive sense RNA from it. You can't do that. You can only take a positive sense RNA and make a negative sense RNA from it. And then from that negative sense RNA we see here in step 4, it can make more positive sense RNA from it. Some of those positive sense RNA are further going to go back to be translated to make other proteins. So in this case, they make nucleocapsids, which are the proteins that actually bind to the RNA genome. Now, the RDRP, along with the host cell proteins, will use the positive as a template to synthesize blah, blah, blah. I already explained that. And the negative sense can then, in turn, be used. I already explained that as well. The viral RNA structural proteins and envelope assemble and are ready for dissemination. So some of these proteins are going to be made right in the cytoplasm, right in the open space, if you will, of the inside of the cell, whereas others are going to be assembled in the rough endoplasmic reticulum, which is basically a place where most membrane-bound proteins are synthesized. So they're going to synthesize those proteins there. These other proteins are going to be assembled outside, and so it's going to attach to the genome of the virus. All those things come together. It's going to invaginate inside the endoplasmic reticulum, and inside and then the Golgi apparatus is going to make another vesicle that it's going to surround itself with. That vesicle is going to fuse with the cell membrane, and then it's going to release the virus. I know, that's all very, very complicated. It's okay if you don't fully understand. You can read it over again, or you can ask me questions later. But this is a basic mechanics on how these viruses replicate. Now, how does it replicate itself? Some more. This is just an image of its entire genome. This genome is you might think is quite small. This is actually very large for an RNA virus. It's about 30,000, well, it says base pairs, but that's incorrect because it's an RNA molecule, so they're not pairs. So it's actually 30,000 bases or 30,000 nucleotides. But whatever, we won't fault them for that. But something by comparison, the human genome is about 3.2 billion base pairs. So it's quite small compared to human, but compared to other RNA viruses, coronaviruses actually have very large genomes. You can see that this genome, so this is basically all of its RNA here, and the red blocks actually represent proteins. So this is where the very start of it is where the ribosome will attach and where it will start translating and making a protein. ORFs are basically a very long portion that is translated all in one, but contains multiple proteins. The proteins are just cut out of themselves. So basically it comes out as one big protein that gets cut to make multiple proteins. So that's what ORFs are. All right, so very, very few things here. But from this, we can learn a lot, actually, about the virus and how it attacks our body. These are some very pretty electron micrograms that have been digitally enhanced to kind of give a more 3D feel of what they look like. So the yellow blobs that you see, the little yellow blobs, those are actual viral particles of SARS-CoV-2 actually emerging from the infected cell, which is the big blue-pink thing in the background, and the same thing on this side. So very cool, but also kind of disturbing. So how does the virus fully take over? So we know that it goes inside the cell and is able to replicate itself, but why doesn't the cell fight back? And that's a good question because cells do have the capability of fighting back all of themselves. There's a thing called innate immunity, and part of the innate immune system is that all cells have it. They have things that they can use to attack any foreign matter that tries to take them over. But a lot of viruses have tricks up their sleeve, and SARS-CoV-2 is no different. So many pathogens produce what are called virulence factors. So I had mentioned those briefly before. It's one of the proteins that SARS-CoV-2 was making. Molecules that increase the pathogen's infectivity and virulence. All right. So SARS-CoV-2 has 3 identified virulence factors. That doesn't mean that it doesn't have any more. These are just 3 that we're very confident it does have. And those have been identified using genomic analysis. So basically, we look at the sequence, we're like, yeah, we recognize these. We know these to be virulence factors because we know that they're virulence factors in other viruses, and especially other coronaviruses. So one of them is NSP1, which interacts with the host 40S ribosomal subunit to induce host mRNA degradation, as well as inhibiting type 1 interferon production. So essentially what this means is, remember how I said that the viral genome, the positive sense RNA, actually has a higher affinity to the ribosome than the regular host cell's mRNA? So that's true, but on top of that, any mRNA from the host cell that does get through, well, now there's this enzyme that's out here that is an RNase. So RNases are enzymes that will break down RNA. And so any RNA that doesn't belong to the virus that comes to the ribosome, it'll be like, nah, I'm going to cut you up. And now you don't work. Now it also inhibits type 1 interferon production. That is very, very important for activating other parts of the cell's innate immune system. So often what happens is it will start to detect viral RNA or other products, and it will release interferon 1 outside of its body, and then the interferon has to bind on receptors on the outside of its body, outside of the cell's body, to be able to fully activate the cell's innate immune system. Without the interferon 1 being produced, those functions are highly limited. And it will also take longer to actually send signals to other parts of the immune system. NSP3C is another virulence factor, and it binds to the host ADP ribose, preventing it from interfering with viral RNA translation. So yeah, ADP ribose is basically a product that our cells make that help block viral RNA from being translated and making viral proteins. But NSP3 actually binds to that instead of having ADP bind to the viral RNA. So that means the RNA is free to be translated and make proteins. And then the last one is ORF7A, which binds to the host BST2, deactivating its function and inhibiting the release of new viral particles. So BST2, it's another thing that your cell has that tries to identify cellular products that are about to be disseminated outside of the cell and stop that from occurring. But this virulence factors will bind to that protein that tries to do that, preventing it from actually inhibiting the virus from spreading. So very, very useful tools that the virus produces to enable itself to replicate and fully take over the cell's functions. How does our immune system fight back? So I have a fun little GIF from this great anime on Netflix, Cells at Work. Super fun. We see immune cells fighting back viral infected cells. Just a note, actually just going back here a sec. All of these things that I mentioned here, a lot of these things are also very common in SARS-CoV, the first SARS-CoV. But they do act very differently in other ways. So there's clear that there's some stuff that we still don't know about SARS-CoV-2 in terms of its virulence factors and how it affects the immune system. All right, so how does our immune system fight back? So the exact kinetics of the immune response to COVID-19 are still not fully understood, but there's a lot that we can infer based on how our bodies and our immune systems have been shown to react to SARS-CoV, and also what we know about other coronaviruses, and in some cases just viruses in general. Some of the things that our immune system does is similar across all viruses. All right, so there is some stuff that we can infer. There's a little bit of data that's come through, but it's mostly anecdotal. So we're still not sure until, there's more research to be done on that, but that may take a while. All right, so as SARS-CoV-2 interferes with regular cell function, eventually the host cell dies through necrosis, releasing thousands of more virions or viral particles. So I mentioned how, the virus essentially stops the cell from producing any of its regular protein. So eventually stuff just starts to degrade. The cell doesn't have any proteins to help make its energy. And eventually stuff just starts to break down. And as the stuff breaks down, the cell just dies. And in some case will lyse very violently. In this case, it's unlikely that it's a explosion of sorts of the cell. It's more of a degradation, but not a very clean one, a pretty messy one that releases a bunch of intracellular material out into the lungs. Now, macrophages and dendritic cells, which are part of your immune system, and those that are in the lungs will phagocytose, so essentially they will eat the virus and begin releasing pro-inflammatory cytokines, enhancing the immune response. So this is a very, very important part of our immune system, signaling our more specialized cells to actually come in and fully destroy the virus. All right, so macrophages, dendritic cells will eat the virus. They'll eat the foreign particles. They don't know it's a virus. All right, macrophages and dendritic cells are just programmed killing machines. If it's not part of the body, then it eats it. It doesn't care what it is. It will do so. but it will release cytokines, all right, which are basically chemical signals that cells use in between one another to be able to communicate. And these cytokines that they release will enhance the immune response. And that's why they're called pro-inflammatory, because one of the steps of the immune system is to create an inflammatory response. And that's why your tissues start to inflame, because your cells are going to start attacking the virus. And in attacking the virus, sometimes there are casualties of our own cells. And, it hurts, but that's how they actually will start to get rid of the virus. These cells will also activate helper T cells, who will subsequently help them in activating killer T cells. Now, these are the big guns. So the helper T cell is kind of like the one who is in charge of directing how the body is going to respond in a very selective, efficient way to kill the invader. And if it's a virus, the helper T cell, which in this case it is, SARS-CoV-2, it is going to activate another type of T cell called the killer T cell. And killer T cells, they're called that for a good reason. They can identify exactly which cells have been infected by the virus. They will bind to them very closely, and then they will release these granules that will cause the infected cell to die. But it doesn't die in as bad a way as necrosis. I'll explain that in a sec. So this killer T cell, highly specific, only targeting infected cells, starting them to undergo what's called apoptosis. So as opposed to necrosis, which is messy and releases a bunch of stuff that the body doesn't want into the lungs, into the outside of the cell, apoptosis is a very regulated, structured form of programmed cell death, in which things are broken down into little pieces and are sequestered away so that the cell will slowly die and lose its function without damaging surrounding tissue. Helper T cells will also activate B cells. These B cells will differentiate into antibody-secreting plasma cells. So, antibodies, very, very important. These antibodies will coat the virus, preventing them from infecting other cells and facilitating their phagocytosis. So now we have the big guns coming in. We have the T cells and the B cells. The B cells becoming plasma cells after the helper T cell is like, okay, do your thing. We'll create antibodies that are specific to this virus. And it's been shown that in other coronaviruses that the spike protein is typically the protein that the antibody will choose as its antigen. All right. So the antibody recognizes this antigen, this part of the virus that sticks out. It will bind to it, and so many of the antibodies will bind to it, then it's no longer able to connect to the ACE2 protein that's on our alveolar cells. It's no longer able to infect us. So the virus is left inert. It's harmless at this point. And also, the back ends of the antibodies are a great place to connect for cells that have the ability to phagocytose, like macrophages, and it will make phagocytosis much more efficient, much quicker, and much more specific. All right, so that's how our bodies fight back, and that's how eventually we clear the infection. All right, this is also very similar to what you would see with lots of viruses, especially other kinds of coronavirus. Okay, so how do we test for COVID-19? How do we know that you have it? Well, you could ask Justin. He unfortunately was chosen as a subject. Well, I mean, he volunteered. He almost insisted on it to get tested. And what they do is they take a sample from a nasopharyngeal swab. So this image here shows just how awful and deep that swab really goes to really get in there and hopefully catch some viral particles. Then we can detect the virus through a method called real-time Rt-Pcr. So what we're actually detecting from the virus isn't the virus itself. We're actually detecting the RNA, so the genome of the virus. So basic explanation how that works is RNA is reverse transcribed into DNA. All right, that's mostly because DNA is much more easy to work with and much more easy to amplify. So RNA reverse transcribed into DNA. It's called reverse transcribed because normal transcription is DNA into RNA. This is the reverse RNA into DNA. So we use an enzyme to do that. And then we continue to do that, and the DNA is amplified. You can imagine that viruses, even just one virus with one strand of RNA, would be pretty hard to detect. That's just one molecule. So we have to amplify the signal so we're actually able to detect it. And then there's a fluorescent probe that's added to the sample. And when the fluorescent probe binds to the double-stranded DNA, it will emit light upon excitation. All right, so it's fluorescent, so we shine a light on it, and then of the right wavelength, it will release a light of a different wavelength, and then we can detect it. And if we get that detection of light, then congratulations, you have coronavirus. There is currently no readily available antigen or antibody test. That is in the works still. It will likely become more readily available soon. They have been able to test and find antibodies for coronaviruses in other animals that they've tested with, like macaques. They've also done tests with humans now, and they have shown, yes, we can identify the antibodies with success. We just don't have it readily available for everyone yet, but that's coming. What is the incubation period? So the incubation period is the period of time before you start showing symptoms. So symptoms typically appear within 5 to 6 days after infection. However, the incubation period has been shown to vary between 2 and 14 days with a very small number of outliers. And by small number, I mean like in the tens of but there have been some people who have claimed, some hospitals and some parts of the world have claimed that people that got infected came into contact with someone, and then about 23 days later, they started to show symptoms. Or even 27 days later was the longest that they showed. But those are outliers. It's the likelihood of that happening to anyone is very unlikely. And whether or not it's true or not, well, that's hard to say. But between 2 and 14, that we know for sure, with 5 and 6 being very, very common. With those with severe symptoms, the time from when they needed mechanical ventilation and when their symptoms first appear is typically 8 days. So if you've had symptoms that were kind of mild, or were getting worse, when it starts to get really bad and you need to be put on a ventilator, typically that's the 8th day after you've started showing symptoms. There are some people who have shown to be completely asymptomatic or otherwise very, very mild and subclinical. Subclinical meaning that a test usually comes back negative, but then later might show positive. So I said here anywhere from 18 to 60%. Now there are experts who say that it's likely around 40 to 50%. But there have been so many different research groups and calculations done that it's really, really hard to say. And so we're still not positive. That's going to be coming as well as time progresses and more research is done. But yeah, 40 to 50% is kind of likely. What are the symptoms of COVID-19? The colors I chose for this are terrible, but that's fine. You can still see enough. So common symptoms we see here: headaches, nasal congestion, sore throat, coughing, coughing up sputum, so mucus basically, shortness of breath, pain in joints or muscles, chills, nausea and vomiting, diarrhea. So that's interesting because at first they weren't reporting some of these things. And I have a table here that kind of shows the frequency at which those kind of less common symptoms appear, because the very common symptoms are fever, dry cough, and fatigue. So fever and dry cough, we see very, very common. Fatigue, still pretty good. Sputum production, still pretty good. Loss of smell and taste. When it first came out, it had been reported that it was about 60%, and it was shown that that was way exaggerated. But there are still studies that show that it might be as many as 30% of people experience a loss of taste or smell, but they usually regain it pretty quickly after the symptoms subside. Shortness of breath, that's when you want to start monitoring your symptoms and really want to check up on yourself and make sure that they don't worsen. Muscle or joint pain, I've had that. It's pretty sure I've caught COVID-19 as well, and I definitely had muscle and joint pains, sore throat as well. Headaches, I know people who've had headaches. Chills, I've had that. I know people who've had that. Nausea and vomiting, that was one that they weren't reporting a lot in the beginning because only about 5% of people have been showing that. But I know some of the members in my household have definitely had that nausea. Nasal congestion, diarrhea. Diarrhea, 3.7 to 31%. Basically, the variance is very hard. It's very hard to say where diarrhea comes from. It could be from a number of things. Could be caused by corona directly. Could be caused indirectly because you're feeling sick. It's kind of hard to say. But the reason we're actually seeing cases of like some gastric distress is because I mentioned ACE2, the protein that is on our alveolar cells. So it is mostly found in cells in our lungs, but it's actually also found in a pretty fair abundance in cells in our intestines. So coronavirus can go in there as well. Oh, that's a good question. So my roommate Alex asked, are the rare, quote unquote, rare symptoms maybe shown as less common because those people go to the hospital less frequently? Absolutely. Because of subclinical patients, these numbers are likely to change. Like the really big stuff is probably pretty close, but it's probably likely that there are people who are like, oh, I'm nauseous. Well, that's probably not COVID-19. But there are some people who you might just get nauseous and might not get a fever. You might not get a dry cough. It's probably unlikely, but yeah, I could see these numbers being higher than they show. And that's likely for a lot of things that we're going to look at in a bit. What are possible complications of COVID-19? So we talked about the symptoms and even some of the uncommon ones, but now what if the shit gets real and gets real bad? Well, you can get pneumonia, which is inflammation of the lungs, which is bad, makes it hard to breathe. Sepsis, which is basically once you start getting stuff into your blood, then you start getting a very strong, so by stuff, I mean like bacteria start to get into your blood. You will get a very strong overreaction of your immune system, and that can lead to sepsis, which can cause a lot of problems. Multi-organ damage, same thing. Your body starts to get attacked by its own immune system. Abnormal clotting, long-term sequelae. Sequelae is basically like after you've had an infectious disease and you're clear of the infection, it's getting damage that is like permanent even after the infection has been cleared. There was the hospital authority in Hong Kong claimed that about 20 to 30% of people that had more severe cases and who were hospitalized after once they cleared the infection, they were fine. They had lost some lung capacity. Sorry, I got that backwards. It's actually that most of them actually lost some lung capacity, and it was about 20 to 30% of lung capacity. I think that's correct. I'm going to double check that in a bit. And then it can, if it gets even worse, it can cause what's acute respiratory distress syndrome. So I have some x-rays and some CT scans here. So the x-ray, so this x-ray is from the same person before and after they got better. So when they have this acute respiratory distress syndrome, basically like severe pneumonia, you can see that it's quite hazy up here where the lungs are, right? Whereas here, it's quite a bit clearer. So those like that patchiness, that opaqueness, represents damage or inflammation. Down here, this is the same. This is for the same not the same person up here, but both of these CT scans are the same person. It's actually the same CT scan. It's just one of them has been enhanced, so you can kind of see it a bit more clearly. But you can see here, this is when after like the onset of symptoms of one patient with COVID-19, it started to worsen. They took this CT scan, this cross-section of the lung, and you can see these patches over here, these white patches, which is consistent with damage of the tissue in the lungs. So this can cause respiratory failure. This is when you're going to be put on a respirator. Septic shock, which is basically when sepsis gets so bad that your blood pressure just drops and you're at risk of just dying. And multi-organ failure, not just damage, but like your organs are starting to shut down. So what causes these complications? Well, it's suggested that in severe cases, SARS-CoV-2, similar to SARS-CoV, is able to decrease helper and regulatory T cell activity. A lot of this is very speculatory. There's only a little bit research on it so far, but we're pretty sure it acts very similar to SARS-CoV. The difference is SARS-CoV, so the original SARS, was much, much better at inducing this severe response. The mortality rate for SARS-CoV is much, much higher than SARS-CoV-2. So be thankful for that at least. But SARS-CoV-2 is better at infecting people than SARS-CoV, the original SARS-CoV is. So, give and take. So we think it's pretty similar to SARS-CoV, but is just not as good at it, at least in most cases. So it decreases the helper and regulatory T cell activity. So what does that do? So without helper T cells, the acquired immune response, which is the immune response that is specific to this particular pathogen, to this specific virus, is very limited in its capacity to clear the infection. And without regulatory T cells, that's another type of T cell I didn't mention before. The immune cells can keep producing pro-inflammatory cytokines. Regulatory T cells are basically the cells that are like, hey guys, let's chill out, okay? They tell the neutrophils and the macrophages and dendritic cells like, hey, you're making too many cytokines, okay? We're already here. We hear you, all right? So it's basically like the shusher, like if you imagine cytokine production as the cells yelling the virus to death, but also a lot of surrounding cells, then these guys are there like being like, shh, it's okay, we're here, we hear you, we're here, it's okay. But if SARS-CoV-2 in some cases will actually decrease the amount of activity of these regulatory T cells, then the other immune cells will keep producing these pro-inflammatory cytokines, which will increase inflammation and increase damage to the surrounding tissue. So the abundance of cytokines attracts neutrophils to the lungs. So neutrophils are the most abundant type of white blood cell in your body. And once they get attracted to the lungs, they will begin attacking in just like a full-blown crazy assault on everything. They attack indiscriminately. So unlike the helper T cell that helps the killer T cell be like, hey, just kill these ones specifically, the neutrophils come and are like, and just go crazy and just destroy everything in their path. This leads to extensive damage of the lungs, eventually allowing for opportunistic bacteria to enter the bloodstream. So imagine that more and more cells are dying. There's so much scar tissue and damage to the lungs because these white blood cells are attacking them so much that it exposes blood cells. to the outside environment, to the open air, and to which allows bacteria that live normally inside our respiratory tract. We have lots of bacteria that live on our skin and our guts and stuff, and they're totally fine. We're not bothering them. They're not bothering us. But now they see an opportunity. Okay, so that's why they're called opportunistic. Opportunistic parasites, opportunistic bacteria. They will enter into the bloodstream because they see an opportunity. They're like, oh man, there's a lot of food in there, and that's a great place for me to replicate and make more of myself. So I'm going to go in because the protective wall of epithelial cells has just been completely destroyed. So it's going to enter your bloodstream, and that's what's going to cause sepsis. And since the body has been fighting the viral infection for weeks, it is now severely depleted, allowing the opportunistic infection to propagate easily, eventually killing the host body. So that's one way that it can kill you. It's either your immune system has been completely depleted, bacteria take over, you die from that, or the other way around, where from sepsis, you go into septic shock, and then you die from just an overload of your immune response. Either way, not great. So how do people get severe symptoms? So why is it that some people get mild to no symptoms at all, and some people, have to be hospitalized, and some people die, no matter what we do to them? Well, a big factor appears to be old age and immunosenescence. So being old, not great. It's not great because, well, when you're old, you're more likely to have a lot of pre-existing conditions that make your body weaker to infection. But on top of that, you go through something called immunosenescence, which is something that we're learning more and more about, as time goes on, as research progress. But essentially how it works is your immune system, especially your T cells, start to go down in efficiency. So I mentioned earlier how SARS-CoV and SARS-CoV-2 probably causes a severe symptoms by working on decreasing the amount of helper T cells. Well, immunosenescence, you've kind of already got fewer T cells to begin with. But you have a robust immune system. In some cases, you still can be producing lots and lots of neutrophils, of macrophages. Maybe this is just my own hypothesis. Maybe it's to try and counterbalance the fact that you have fewer of these effector cells. I'm not sure. But essentially, you start to lose that specificity. In some cases, you might even lose your immune memory. So when you get an infection, a lot of infections, typically your body will keep some T helper cells, some killer T cells, and some B cells that remember, I've seen this virus or I've seen this bacteria before. And if it tries and comes back again, I'll eliminate it in a snap. And, that's how immunity works because of these memory cells. But in some cases, you start to lose your cells start to lose their memory. So that's why it's called senescence. Your cells start to go senile. So we think that could be a contributing factor. Another thing is high viral dosage. So there have been studies that show that in coronaviruses, the more viral particles you're exposed to, the greater the severity of the symptoms. That's been done with animal models. There's no evidence for humans because that's unethical. There's no scientific institution will get the funding to test on if we can give more of a virus and infect people, if we can make them so sick they'll die. So, we kind of have to, go by animal models on this. So it's a possibility. It hasn't been tested on animals with SARS-CoV-2, but it has been shown with quite a few endemic viruses to their specific species. Pre-existing conditions. So I already mentioned how old age, when you're older, you typically have more pre-existing conditions like diabetes or heart disease or cancer or what have you. If you're pre-existing conditions, then your body is already weakened and is therefore more susceptible to damage from a foreign invader. Suppressed or compromised immune system. So Suppressed immune system, typically by certain drugs, can suppress your immune system. There are a lot of diseases that are autoimmune diseases where your immune system is already attacking specific parts of your own body. And there are lots of different kinds of medication for that. There are people with Crohn's disease, for instance. There's medication they take to reduce your immune response to your gastrointestinal tract. But in reducing your immune response there, you could be reducing your overall immune response, therefore making you more susceptible to infection. And compromised immune system, so like someone who has HIV/AIDS, for instance, obviously going to get hit quite a bit harder by something like SARS-CoV-2. Being male. This is quite interesting. I'm going to be showing some stats in a bit that kind of help justify that. Now, the number of people who are infected, it's usually pretty close to 50-50. But for serious symptoms, males are more likely to get more serious symptoms than women are. That's not uncommon for a lot of stuff. Having one whole other chromosome, if you're female, can really help. Unhealthy lifestyle. Yeah, if you're just generally unhealthy in your life, then, your body's not going to be as well prepared as a healthy body. And then there are probably likely other unknown environmental and genetic factors that, could take years and years of research to try and figure out and see what they mean. But, for the most part, this is pretty good. All right, so this is a graph that I made, percentage of case severity in Canada. So this is based off data from the Government of Canada website, and I got it today, so it's as up to date as it can be. And essentially, 95% of people who catch the virus will have mild cases, and 5% will get serious cases. So 5% might not seem very high, but 100 people isn't a lot, and 5 out of 100 people catching it, and you multiply that multiple times, it racks up. So how do people get severe symptoms? Well, in Canada. So I have this graph. So this graph comes again from that website, the Government of Canada website. It takes into account 256 patients who were hospitalized, that had to be hospitalized. All hospitalization data, however, is available for only 2,036 of all cases. They said that it was because they had a limited sample size just because these were the patients that they had the most data on. Like, they had a lot of information on each one of the patients. So they wanted to take that pool of data. They wanted to take those patients that they had a lot of information on and use that data to be able to make these graphs and everything, instead of taking from, perhaps a larger group, but with, less data. Anyway, just wanted to specify that. So in Canada, based on this data, 58% of hospitalized patients are above or equal to 60 years old. But you can see in this graph here, so male and female, you can still see that there are quite a few people in their 30s and their 40s who, are still getting sick and needing to get hospitalized. Mostly for men, but there are still some women as well. But you can see that the older you get, the bigger it gets as well. I mean, it gets lower here because there are, fewer of them. 61% of hospitalized patients are males, mentioned that. 64% of hospitalized patients had one or more pre-existing conditions. So that's important to note that so a lot of them did have pre-existing conditions, but 36% that didn't have any pre-existing conditions and still needing to be hospitalized is still pretty big. So there's obviously still a lot that we don't know about what leads to the severe cases. And 1% of reported COVID-19 cases have died in Canada. So 1% is lower than the world average that I'll show in another graph later. So it's looking okay so far. Obviously we don't want any deaths, but if we can't avoid them, mitigating them and making sure it's as low as possible is still very good. Globally, in Italy, actually, there was this one hospital foundation that actually reported that 88% of deaths in all of Italy due to COVID-19 were accompanied by one or more comorbidities. So other conditions that they were also sick with. I find that's quite high. So I don't know, I'm maybe a little skeptical about it, but another report came later that corroborated and actually even had a higher number. The only reason I'm skeptical is because Italy, so many people we think are also dying because they have lack of access to actual facilities and actual medical equipment. But anyways, that's what was reported. So I put it in there. Take it as you will. The estimated global case fatality rate is between 1.4 and 2.3%. So it's probably more on the higher side currently. So we are still under that, like I mentioned. So that's good. Canada's doing okay so far. What is the fatality rate? So this is a graph. This is based on data from China. So again, take that with a grain of salt because China has been really, really bad in terms of silencing anyone who has anyone who would critique the government's response to the crisis. There's some shady stuff coming out from people who are reporting and keeping their names anonymous and everything. So who knows how big the numbers might be, but this is what we got so far. So again, you can really see that the older you are, the much more likely you are to die from complications because of COVID-19. So that's important to keep in mind. That still doesn't mean, again, there's still 0.2%. And this is just for China. There's no data I could find available from the demographics, the entire amount of people who got sick in Canada. I think they're still working on that, or I just didn't find it. But so hopefully we'll get that in the near future as well. But it's still important to note that, the older you get, the more likely are you to die. We've all heard that. But keep in mind that, if you're younger, still be careful. How long does it take to clear the virus? All right, so this is kind of interesting. Mild cases typically recover after 2 weeks. Severe cases may take 3 to 6 weeks to recover. And for those who have died from the infection, the time between onset of symptoms and death varies from 2 to 8 weeks. All right, so usually there's quite a bit of time from when you were infected, when you start getting symptoms, and when you actually need to be hospitalized, and when you unfortunately pass away. All right, this is just a happy lady who's like, yeah, I'm free, I don't have COVID and blah, and now allergens. I don't know. I thought it was cute. How is COVID-19 treated? One sec here. All right. How is COVID-19 treated? So in most cases, I mentioned 95% of cases in Canada pretty mild. In most cases, those infected will clear the infection themselves with lots of rest and liquids. Acetaminophen can mitigate symptoms. Now, there was initially a report that said that ibuprofen should not be taken because it will actually worsen and create complications if you have COVID-19. That was tweeted by the Minister of Health in France, and it was because of some anecdotal evidence. So we're not exactly sure if that's really the case. The WHO actually, they actually reported that it was true at first, but then they retracted the statement and they said that there was no significant data to show that it was true. But still, stick with acetaminophen just in case, whatever. In severe cases, patients may require mechanical ventilators, fluids, and antibiotics. Antibiotics, because like I mentioned, if you get sepsis, then you have bacteria that were in the inside of your lungs that are actually going to get into your blood vessels and into the rest of your body. So you need to go on some pretty strong antibiotics to get rid of those if you want to survive. There are no antiviral drugs specifically for SARS-CoV-2 as of yet, but there are various kinds of treatment that are currently being tested or developed. So there are a number of proteins that are considered candidates if drugs are to be developed. There are quite a few targets for it. So any ones of the virulence factors that I mentioned, so drugs that are produced that can deactivate those virulence factors of SARS-CoV-2 would be great. Any medication that will bind to the spike protein and prevent it from actually entering your cells, that would obviously be great. But here's some data, some stuff about some treatment that they're considering right now. So nitazoxanide, which is a wide-spectrum antiviral, has been recommended for further testing for its effectiveness at treating SARS-CoV-2. Currently, there have been no tests on that, but there have been some doctors who have recommended testing with that. Tocilizumab, whatever, is a immunosuppressant, which could be useful for treating the cytokine storms in people with COVID-19. Cytokine storm basically just means, so like when I mentioned that, you get an overreaction of your immune response and your body just is full of inflammation and your immune system starts to basically destroy your body. That is called a cytokine storm because there's just so many inflammatory cytokines in your body. So this drug is an immunosuppressant. It will bind to interleukin-6 receptor, which has been shown to be a very, very important pro-inflammatory receptor for actually inducing that kind of inflammatory response. It's shown to be effective as an immunosuppressant, so it could be useful in treating people who get these cytokine storms because of complications of COVID-19. But testing with that is still ongoing, and I'm not even sure at what stage of it they are. Chloroquine is an anti-malaria drug that has some antiviral properties. So you may have heard of this from the news because Trump was all like, yeah, this could be a game changer, blah, blah. And then it started going off the shelves and people started taking it and overdosing. It's still in trial for its effectiveness against COVID-19. It has been shown in the past to have antiviral properties, interfering with its ability of replicating its RNA. But the tests for, how it will be effective against COVID-19, against coronavirus, is still ongoing. So we still don't really know for sure. I should mention that this drug here is actually in phase 2 of testing, which means that they're testing for possible side effects when given to patients. This one just started clinical trials actually on March 24th. So yeah, it's still pretty new. Remdesivir is an antiviral drug developed to treat Ebola virus, and it has been considered a strong candidate for treating SARS-CoV-2. It also interferes with RNA virus replication with the actual machinery. But and it is currently in phase 3 of testing, which means now they're testing how effective it is at actually treating SARS-CoV-2. So it's the one that is in the furthest along with testing on patients. So we'll see. Hopefully news from that will come out pretty soon. Some companies are working on developing and testing passive antibody treatment. So one of my roommates, Johnny, posted on our house Facebook page about how Mount Sinai Hospital here in Toronto we're looking for volunteers for people who have caught COVID-19 and who have cleared it to be able to give some of their blood so that they can try and give some of the antibodies that we've produced to patients who are in need of treatment. Now, these kind of treatments have been shown to be effective in some cases with other viruses. Usually, it's much more effective very, very early in the stages of symptoms. So if you've had symptoms for a very long time, typically, I don't know about the tests that they've done now with human. I don't even know if they've really done many tests with SARS-CoV-2, but with other viruses, it's usually like the earlier you get it done, the more effective it is as a form of treatment. Because technically, it's, you would still need to produce your own antibodies, and anyway. But there's other drugs that have been tested and people are looking into. There's actually they tried some HIV medication, which was shown to not be effective at all, which is not really surprising because that drug was developed so specifically for HIV. So it kind of makes sense that it didn't really work for SARS-CoV-2. Now, how does it spread? How does COVID-19 spread from one person to another? So people are contagious when they are symptomatic, likely 2 to 3 days before showing symptoms. That has been shown fairly consistently now. There have been some skeptics out there, but most doctors and virologists now are pretty sure that 2 to 3 days before you start showing symptoms, you are shedding viral particles, you are infectious. And possibly a few days after symptoms are cleared, that's not been shown 100%, but some people do suggest that it's still a possibility. Then people who are asymptomatic are possibly contagious as well. I said possibly, but now it seems very, very likely that they are. There were some reports that were showing that they were people who were asymptomatic. The first group that said that people who were asymptomatic were contagious, that initial research was shown to be wrong because they hadn't interviewed the person who actually spread the virus, and they assumed because everyone else there was like, oh, well, she was fine, she wasn't sick. But then this other group actually spoke to her and she was like, no, I was sick. I was like taking like medications because I was coughing and I had a fever and whatever. So, people were like, so there you go. You can't spread when you're asymptomatic. However, more studies have come out later and show that it is actually quite likely that that even if you're asymptomatic, you could still be contagious. However, China has recently said, and they don't even report people who are asymptomatic. They claim that if you're asymptomatic, there's no problem, that there's no possibility of you being infectious. But most researchers think that you are now. Transmission occurs primarily through contaminated respiratory droplets upon close contact, like these gentlemen making out over here. That is not social distancing. Or after a sneeze or cough within a 2-meter range. Viral RNA has been found in stool samples of patients with COVID-19. So that's consistent with how I mentioned earlier that the virus can bind to pretty much any cell that has an abundance of ACA2 protein. And because there are cells like that in your small intestine, it would make sense that you would find some virus there. So, but it's still unsure if from fecal matter, if it's able to be transmitted. We know that we can detect it with Rt-Pcr, but still not sure if it's contagious. Contaminated surfaces could possibly contribute to transmission given how, I didn't show that there, but given how long the virus can survive on surfaces. And also, Yeah, well, I'll show you in a sec, essentially. So how long can the virus survive outside the body? So this is based on a study where they looked at a bunch of surfaces, they put some virus on it, and then they saw how long they could detect it after initially exposing it to viral particles. So first we'll look at the detected, and then I'll explain what I mean by estimated. So aerosols, I know that's not a surface technically, but they also tested for that. So they showed that when sprayed out into like a mist, like as if someone sneezed, viable viral particles could be detected within 3 hours. Copper 4, carbon 24, and longest was stainless steel and plastic for 72 hours, which is very, very long. And this is not just RNA, this is viable viral particles. So alive viruses that have the potential to infect someone. Now, estimated. The reason that they also did some calculations and some statistical modeling to extend the length at which the virus can survive here is because detection of live viruses is kind of limited in the number of viruses that need to be present in order for it to be detectable. So it's possible that most viruses have died and it's no longer detectable, but they think that it's likely that there are still some that are alive that are there. So through their estimations, through their calculations, they think that it could be up to, I couldn't find anything for aerosols, but for copper, they think it could be up to even 18 hours, 55 hours for cardboard, stainless steel 90, and plastic up to 100 hours. So yeah, cleaning surfaces, good idea. Note that viral RNA found in multiple surfaces of the Princess cruise ship 17 days after passengers disembarked and before disinfection commenced. So in this case, they didn't test for viable viruses. They only test for the presence of RNA. So it could be just fragments of, or just attenuated viruses that are no longer infectious, dead viruses in the environment, but they found it on quite a few surfaces. And RNA, I've worked with RNA before in the past. I worked in an RNA lab, and I can tell you that RNA is typically very, very fragile and degrades very quickly. There are RNases, so proteins that degrade RNA all over the place, and RNA is fragile, single-stranded, usually degrades pretty fast. Who knows, maybe those proteins that are around the RNA, the nucleocapsid, maybe it's pretty good at protecting it. You know, who knows? But anyways, all of this to show that this doesn't necessarily mean that, after 17 days, there were live viruses on these surfaces. But it's still interesting to note. So how do we stop it from spreading? So look at this little kitty. Oh, he's trying his darndest to just clean the whole place. Just make sure there are no corona. But he misses the toilet. Poor thing. So wash your hands frequently and correctly. That is very important. The other thing that is important is social distancing. This has been shown consistently to be the best way of preventing the spread of the illness, because it appears to be so contagious that keeping away from other people just is the best way to make sure that you don't catch it. We all know this now. We've listened to the news a bunch. Physical distancing, so even when you are with people, stay 2 meters out of reach at least. Clean surface frequently. As we saw, it can live on surfaces pretty long. So make sure to clean the surfaces. You should note that washing your hands with soap and cleaning surfaces with soap, very good at destroying the virus, because the virus is enveloped, which means it has a phospholipid bilayer. That's basically can be removed with soap. All right, it will react with the soap. The soap will just slosh it off, and then the virus no longer can infect. It's just a bunch of, harmless RNA particles. Avoid touching your face. That's how it likely gets into your body, is through your eyes, your nose, your mouth. So avoid touching your face as much as possible. Cough and sneeze in tissue or your elbow. Masks. So you might see sometimes a lot of people wearing masks, just like walking around outside. Most experts actually agree that you shouldn't wear a mask unless you're coughing and sneezing persistently, or you're a healthcare worker and you have full personal protective equipment, PPE, on you at all times. Because if you are just a normal person who has a mask, chances are you're putting it on, taking it off, touching your face a lot, and you're actually more likely to get the virus. So yeah, only wear a mask if you're coughing and sneezing. Vaccines are currently only in preclinical trials. So there are, however, fortunately, many companies who are working on making vaccines. Some of them are trying cool and new innovative things. There's this one. There are some that are trying to develop RNA vaccines, which basically means that they're going to put a strand of RNA that is pretty similar to the RNA genome of the virus, put it in a nanocapsule, put it into your cells, into your body, then your cells absorb them, and then it will produce some of the viral proteins that, SARS-CoV-2 produces, but it won't be infectious. However, your body is then able to make an immune response to those foreign proteins and therefore produce antibodies that will protect you if the virus does ever come to you. Like, I've seen those proteins before. They were by themselves then. Doesn't matter. It's bad. Let's destroy it. So instead of the regular vaccines from before, where it's you make the antibodies, or you basically put an attenuated virus or a dead virus or something along the line, put it in your body, and then the body makes a response according to that. You put the RNA and your body makes the proteins itself, and then you get an immune response from that. You get immunity from that. That might sound a little complicated explaining it this way. If you have any questions later, you can ask them and I can explain it a bit more detail. But essentially, it's just a really cool new way of making a vaccine. Oh, we do have a question. Body? Grant, why do you want me to say body? Do I say it weird? Do I sound like a maritimer or a French person when I say it? It's fine. And last thing, governments have declared a state of emergency to try and combat the spread of the infection with varying degrees of success. We have South Korea that's done very, very well. I said it a lot. Okay. That's fair. I do tend to ramble when I'm nervous. I'm so nervous, you guys. So yeah, governments have declared a state of emergency. South Korea has been super successful in stopping the spread of the virus. Canada, well, that remains to be seen. Hopefully we're doing pretty well. Italy has done terribly, and United States has done also terribly. So we'll see, but closing borders as much as possible, keeping people working from home or laying people off. These are all shown to be pretty effective strategies for stopping the spread. So why should we care? Whatever, who cares? So I'm sure you all heard on the news this whole flatten the curve thing. And although you might be kind of annoyed hearing it because you've heard it so much right now, it is a really smart thing to do. Essentially, the problem is if our healthcare system becomes so overburdened with patients that require care because of COVID-19, then eventually there won't be enough beds, there won't be enough healthcare workers, there won't be enough equipment, enough medicine, what have you, to be able to treat the patients who would survive otherwise. And in that case, we will see a sharp spike, a sharp increase in death, like we see in Italy, where it's about 10%. So we want to avoid that at all costs. We don't want our healthcare system to be overburdened. Right now, in some provinces, they're a little strained. I mean, that's not uncommon, honestly, for our healthcare system in Canada, but they are managing. They appear to be quite well prepared for now. So that's very, very good. The problem is also that if we if we go over capacity for our healthcare system for COVID-19, then people who need treatment because of other illnesses or injuries or what have you, in some cases much more serious, much more fatal, well, there's no place for them. We're treating people with COVID-19. If you come in here, you're going to catch it too, and then you probably will die. It's a whole shit show. So we want to avoid that happening. We don't want to have to choose who gets to live and who dies like they are in Italy. That's kind of dark. Now, this is a really important question. Can you catch the virus multiple times? So, this is how it usually works. If you successfully clear a viral infection, then you are typically immune to that strain of the virus, at least in short term. Your body, your plasma cells, typically produce such an abundance of antibodies against this virus. And I think it's immunoglobulin G, those particular types of antibodies that usually stay in your body for quite a while after the time of infection, even up to like a year after infection. So short term, yes, in almost all cases, yes, you will be immune. However, whether or not the virus provides long-term immunity depends on how it interacts with the host's immune system. So it's unknown whether SARS-CoV-2 provides long-term immunity. So I mean like past a few months to a year, if you are immune for longer, like in the case of if you get the measles vaccine. You know, if you get the vaccine, you're immune for measles for life. You can never catch it. But if SARS-CoV-2 is like that, we're not sure, but other coronaviruses don't. I can't remember specifically for for the original SARS, but I know for rhinoviruses like the cold, you can catch the same strain of the cold even a year later after all of your antibodies have gone away. That means that basically there are no memory cells that were made. So memory cells are the ones who, after all the antibodies that you had made originally were gone, these cells stick around and will elicit a powerful, quick immune response if that infectious material ever comes into your body again. But in some cases, depending on how the immune response acts and how it reacts to and interacts with the virus, sometimes memory cells aren't produced at all. And that has been shown with some viruses, especially coronaviruses. So, you know, that might be hard to say. 5 to 10% of patients in China with COVID-19 that recovered and tested negative for the virus tested positive when being tested, in some cases, weeks later. So this is something that we've been hearing about a few times now in Southeast Asia, but primarily China, saying that patients who recovered, they had symptoms, they tested positive, then they recovered, tested negative, came back, and usually they were sick for something else or whatever. They obviously didn't think, oh, it could possibly be COVID-19 again, got tested and tested positive again weeks later. So what's happening there? We still don't really know for sure. It's extremely unlikely that they are recatching the virus so soon after clearing it. Because like I said, especially for the few weeks and few months after you've gotten the infection, your body is full of those antibodies to protect you from that same infectious material. And because the virus hasn't mutated much, it's very unlikely that it's a totally different strain of the virus, especially if they haven't traveled. Presumably, most of them haven't traveled much since then. So most likely even the exact same strain of the virus with almost no mutations at all, it's extremely unlikely. So what's more likely is problems with the testing. There are some whistleblowers outside of China saying that their tests are actually only 30 to 50% accurate. This was more in the early stages. Hopefully they're more accurate now. And also, this is anecdotal. It's not, it hasn't been shown that their tests are that much, but this is someone saying no, I work with the medical team in China, and I'm telling you, it's about 30 to 50%. I think it's about 30 to 50% accurate. So it's possible that, it was a false positive the first time, or it's a false negative when they tested negative that time, and then the symptoms just got worse again. It's really hard to say. It's also possible that when their symptoms got better, that maybe they were less infectious and they were just producing fewer viral particles. That can be common with people who are asymptomatic with a viral infection. Sometimes you are still infectious, but sometimes you're less infectious. So maybe they're just not able to detect enough of the RNA to be able to get it. Anyways, I know it sounds scary, but it's most experts now think that it's actually quite unlikely that you can get it twice. Especially in the next year, it's pretty unlikely. Now, tests done on macaques showed that they produced antibodies against the virus and could not be reinfected. And in fact, now there are tests on humans as well that have come positive with antibodies. So it shows that we do make antibodies. And like I said, if you have a bunch of antibodies in your body, Grant, you are right. I do say body a lot. But if you do have an abundance of antibodies, there's no way you're going to catch that infection again. They are so good at specifically targeting pathogens. And like I mentioned, the virus has not mutated much. In fact, there are currently 410 sequenced genomes of samples of this virus worldwide, taken from 5 different continents. So it's a really big sample size, really big spread out all over the world. And from those, the one that had the highest number of mutations out of the 30,000 nucleotides that make up the viral genome, the strain that had the most number of mutations was 7, which is very, very low mutation rate. So that's good in terms of like there being a second strain hitting us again all over again very soon. Pretty unlikely. So how many people are infected? So here is a fun little graph that shows how the virus spread around the world. So you can take a look at that. It's pretty cool to watch. Now, it was on March 11th that the WHO declared COVID-19 a pandemic. All that a pandemic means, really, pandemic has nothing to do with how deadly or severe an infectious disease is. All it means is it's about numbers and speed. So how fast is this virus growing? This is also totally out of date, by the way. That just went up to February 29th. Brendan asked if low mutation rate is linked to how resilient it is when just surviving on a surface. I haven't read anywhere in which that is the case. I would think it's unlikely. It's more likely a case of just the general structure of the viral particle itself. Yeah, it's just pretty strong. So it is enveloped, but if you look at the actual structure of the virus, you can see that even though it is a phospholipid bilayer, it is coated in transmembrane proteins. So that could be the case that the capsid could prevent. It's not technically a capsid. It's still part of the envelope of the virus. The nucleocapsid are the proteins that are inside the envelope that coat the RNA. It's not like a typical capsid that we see in other viruses where it's like, a cool like geometric shape. It's kind of just like a bunch of proteins that goes around the RNA. But yeah, the protein on the envelopes, the abundance of protein, that could be it. I don't know. That's just a hypothesis. Anyways, so yeah, you can see that this spread quite a bit all over the world and has gotten much, much worse since this graph was made, this little animation was made. So what I was saying is that a pandemic has to do with how fast the number of people, the amount of infection occurs, how quickly it spreads. And a pandemic means that it's worldwide. All right, so we've had a number of pandemics. throughout our lifetime, we've gone through like at least uh, two that I can think of, SARS and H1N1. But neither of those were as bad as this because of its how infectious it is combined with how deadly it is. So it's not as deadly as SARS in terms of how the percentage of people it kills that catch it, but it is much more infectious than SARS-CoV, the original SARS. And SARS-CoV-2 is not as infectious, however, as H1N1. However, H1N1 was far less deadly. It was still pretty deadly, but far less than COVID-19. So it's kind of, not exactly, but it's kind of in the middle of the two. And that special like place there has made it very dangerous and has created this unprecedented event that we're now living. So how many people are infected? A lot. A shit ton. So total number of confirmed cases, it's actually higher than this now, but from my last count is 743,286. This worldwide. And then from those, there are 157 and 75 patients who have been confirmed to have recovered. So there are still quite a few people who are still sick. The basic reproduction number for SARS, so that's the R value for SARS-CoV-2, is estimated to currently be between 1.4 and 3.9. So the basic reproduction number, you may have seen that on the news and stuff somewhat, and you may know a little bit about it. Essentially, it's a value that determines approximately how many people one person who is infected will pass on that infection to. All right, so if the basic reproduction number is 2, then one infected person is likely to infect 2 people. All right, this number has nothing to do with the rate of infection, how quickly it spreads. All right, so it's actually quite limited. It can be used in calculating these things, but by itself, it gives you just a little bit of information, but not quite. So we can see here SARS-CoV-2 is believed to be, the value is believed to be somewhere between 1.4 and 3.9, whereas with SARS-CoV, the original SARS, it's actually believed that it was somewhere around 2 and 4. So actually maybe even a little bit higher there, but like I mentioned, SARS-CoV-2 is clearly much more infectious. Oh, what would be the R value for the seasonal flu? That's actually a really good question. I believe it is quite a bit higher, but I'm not positive. Oh, that's actually not that much higher. Yeah, so 1.5 or 1.3 to 2. Someone posted on our chat thing. But yeah. So again, it's a number that gives you some information, but it's still kind of limited in what it tells you. And it also can vary quite a bit based on basically based on how a population behaves after they know about the infection, and also different environmental conditions like the weather and various climate changes, stuff like that. Like I know that for the flu, you typically see much more cases of it during the wintertime. You start seeing it more in the fall, and it gets worse in winter. And you see fewer and fewer cases during the warmer months. But so we actually don't know if that might be the case for SARS-CoV-2 or not. This is a fun little diagram. It's on a logarithmic scale, linear scale that shows the relation of total number of cases versus total confirmed deaths. So total confirmed cases, total confirmed deaths, logarithmic scale, so goes exponentially in the tens on both axes. You can see here that Italy, quite high, 10%, a little bit above 10% of a fatality rate, and has quite a high number of total infections in general. The United States just beat it. and is now the country with the highest total number of infections. Canada is somewhere down here, I think, or like here. I think that's us. Yeah, dark green, North America. And our death rate is about 1%. And our number of infections is almost 7,000 now. But yeah, so United States, pretty poor planning on their part. Same with Italy. Italy also has, unfortunately, an extremely old population, so not doing so great. Whereas countries like South Korea and Germany have been considered to be some of the best countries to prepare. Germany, they have a high number of beds to hospital beds per capita compared to a lot of other countries. So that's a very positive sign that they are already well prepared even before this became a pandemic, and that they have started producing much more protective equipment and stuff very quickly to prepare in case it became serious. Hopefully Canada will follow suit. In some cases we have been doing very well in terms of ramping up manufacturing of stuff and preparing our hospitals for the potential of many, many more cases. How many people are infected in Canada? So last count was 6,671 confirmed cases. I think that is a bit higher now. And this is a graph showing, and it was accurate up to yesterday or 2 days ago. 114 patients have confirmed to recover. So that is good, but there are still quite a few. Ooh. Oh, yeah. Someone just showed me. It is now 7,474 cases. Yeah, that's high. That's high. So we are getting more and more, which, never fun. The number of asymptomatic and unreported cases is unknown. A lot of people will, if they're subclinical in terms of having symptoms, then, they're not going to go get tested. A lot of the times they'll be turned away. That might change in the near future, especially once we understand more of how infectious people who are asymptomatic might be. And also people who have very mild cases are probably not going to risk going out to the hospital and potentially infecting other people. They might just be advised by health services to just self-isolate. So in some cases, we're not exactly sure the exact number, but, pretty good idea still. How many people are infected in Canada? This is daily new cases. So it's not the total cases, but how many new cases we get every day. So we can still see that we're still pretty high here. Hopefully what we want for the end of this week is that we are going to start seeing a decline of new cases. It makes sense that we're still seeing some high peaks here because it takes so long for the virus, for the disease to start for people to start showing symptoms at all. So it makes sense that we're still going to be seeing some of these high numbers for a few more days. But hopefully near the end of the week, we're going to start seeing lower numbers there. And that will mean that, our measures of self-isolating and everything else is actually helping and is going to flatten the curve, as we've been saying for so long. Are efforts working? We'll know more in the coming week, like I mentioned. This is a graph that shows, essentially, it's like flattening the curve, but how flattening the curve is just a step, and then to eliminate it completely, we need to stop the spread. So essentially, what is important is how long are we going to be putting these systems in place in which we currently have to make sure that no new people get infected. If we do it for too short a period of time, then there might be people who are still sick, who might not realize they're still infectious. Maybe who knows? And they'll start infecting people all over again. And then, we'll be right back where we started, and in fact, perhaps even in a worse situation. So what is the timeline for what would be the correct amount of time to wait? We don't really know. That's still data that's incoming based on how long the virus lasts. Like I mentioned, in some cases, the amount of time for people to actually get symptoms can be very long, between 2 and 14 days in most cases, even though most cases between 5 and 6. So if you just started getting symptoms 14 days and then you're sick for another week or two, that can be a very long time that, you might start spreading the disease. But knowing how infectious you are in those periods of time and those sort of things, that's research that's still ongoing. And based on that and based on other countries and based on statistical models, we'll hopefully figure that out. But right now we still don't know. So what's next? How long will it last? Like this pandemic, we really have no way of knowing. It could probably be at least another month, maybe 2, maybe more. But hopefully with these continued measures and social isolating and everything, it will be sooner rather than later. How serious will it get? Hopefully, again, here in Canada, we feel like we're pretty well prepared and we're doing all we can so far. So hopefully it won't get too serious that it will become another Italy. So that's people seem to be fairly confident. A lot of doctors are bracing themselves for the storm, but still feel like, they're prepared. How long will the preventative measures be put in place? So like I mentioned, that's still not really known. Another month seems very likely, possibly 2 months. Any more than that, we're not sure. Hopefully it won't be more than a month. How will it further impact my life? That's also hard to say. The longer we're away from work and some places that aren't considered essential, maybe some other places will have to shut down if things get worse instead of better. Then that could severely impact our lives. And that's kind of a scary thought, but we'll cross that bridge once we get there, if we ever do, which we hopefully might not need to. What impact is this going to have on the economy? It's already shown that the economy has suffered pretty heavily from this. Only requiring essential services to stay open means that a lot of stuff isn't, and the economy is suffering because there's no flow, or there's a stoppage, and that could create some problems as well. The governments around the world are trying to mitigate that, but the longer we wait, the tougher that's going to become. And that is true. What impact is this going to have on politics? There are actually some reports that are showing that this could create much more of polarizing with politics. So we can see people who become much more liberal, who are like, this is showing that capitalism is bad and we need to tear it all down, blah, blah. And it could show people who are very going to be stuck in their ways. It's like, no, we need to get back on track. We need to get back to work. What can we do? So there are some, basically some political experts out there who are saying that it could create a bigger divide, which is funny because there already is a pretty big divide in politics nowadays. What impact will this have on our ecology? Well, we've seen that lots of places around the world, like Venice, for instance, their canals have been the cleanest they've been in centuries, because of no human activity. So, maybe this is, try and think of it in a positive way. How could this possibly impact the world in a good way? There have been a number of places that have shown a reduction in smog, like even Los Angeles, and parts of China as well have shown that. They've shown there are places where you can see the sky that you haven't been able to see it in years. So that's kind of funny. What impact will this have on society? Who knows? But chances are it probably will create some sort of changes, because you can't go through something like this and not have it affect you in some way. It's pretty big. So stay safe, stay sane, stick to your roots. These are just little pieces of advice that I'm giving you my therapist gave to me, and that you'll find online in some places that try and help you cope with self-isolating and not being at work and everything. Sticking to your routine is a really good idea. So getting up at a specific time, going to bed at a specific time, eating meals at specific times. Chat often with your loved ones. It can be very isolating. I'm fortunate enough that I live in a big house with a lot of other people. Some people don't have that chance. And even then, reach out and talk to other people you care about. Continue with your hobbies or pick up new ones. Work on chores. Hey, you're stuck at home. Might as well clean shit. I still have not gone around to doing that to my bedroom, but I really need to. Try exercising and meditating. Staying indoors a lot and stressing yourself out and just sitting in one place all the time is not the healthiest. But meditating and exercising, good for your mental health and your physical health. Have a good night's sleep, always a good idea. Eat healthy, always a good idea. And just remember, we're in this together, and it's all only temporary. This is supposed to be a heart emoji, and I am furious that it did not show up. But whatever. So that was a pretty long presentation. I apologize. I'm pretty long-winded. I hope you guys learned some stuff. If you have some questions, please let me know. Actually, you have a question, Sasha? You have a gazillion questions? Yeah. This is Sasha, everyone.
Hello. So at some point you were talking about two proteins called NSPC1 and 3. I assume that means there's similar patterns of these proteins because they have the same name but different number.
Yeah, so that is very common with what's called orthologs and paralogs. So I'm not going to go into too much detail about that, but essentially, not only are organisms related to one another, but all genetic material, all genes are related to each other to some extent or another. And genes that are closely related and have similar structure and in some cases similar function, they often get numbered like that. Yeah.
Okay, so would that mean that these proteins that the virus tries to encode for, can these proteins be targeted by a wide-ranging antiviral or?
Yeah, so viruses if you're asking if there's antiviral medication that could target all of that family of proteins,
right?
Absolutely, yeah, that could happen.
That is awesome. Okay, questions about immune system. You were talking about neutralization, opsonization, and the complement system when you were talking about how you recruit antibodies to prevent viral action. This is out of curiosity, but would specific strategies be employed for specific pathogens, or are all three used in any pathogen occurrence?
Sorry, could you repeat which strategies for what now?
So you know how all the antibodies will coat the surface of a virus at some point to render it useless?
Yeah.
Or will coat it and recruit macrophages to eat them, or even recruit proteins to pierce holes in it and destroy it. So those are three different strategies that the antibodies could use.
So the antibodies almost always do those two things of coating the virus and preventing it from infecting other cells, and also because the tail of the antibody is able to be very easily recognized by cells that can phagocytose, like macrophages. So they do those two things at the same time. Right. And that's consistent with all viruses. Okay. The last thing you were talking about destroying host cells, that's not antibodies that does that. It's killer T cells.
Right.
So it's another type of cell.
But they can recruit the complement system, though. They can, the antibodies could also recruit a bunch of proteins to gather and form pores. I'm not talking about the...
Yes, they can. That is something that they can do as well. It's just that typically that kind of happens later, and usually effector killer T cells will be the first line for doing that. Or not the first line, but we'll be doing that more so.
Fair enough. Oh yeah, would an asymptomatic person be at risk of developing severe like symptoms right away? Could like a person be asymptomatic entirely and then tomorrow, oh, your lungs are dead, like, or
Not that fast, but yeah, you could be asymptomatic and then develop severe symptoms.
like right away?
Well, usually, again, it's like the severe symptoms start within 8 weeks of someone, like not 8 weeks, sorry, happen 8 days after someone has gotten symptoms. So probably not, because yeah, that is the case for most people, is that you are asymptomatic, then maybe you get symptoms, and then 8 days later you get it. Am I saying that that can't happen? No, I'd have no idea.
And here's a question about people being asymptomatic. Does an asymptomatic person necessarily resist the virus, or is the virus just there reproducing and not causing the symptoms?
That's a really good question, and it's kind of hard to answer because it sometimes depends on the virus. It could be that your immune system is just really, really good at fighting it. And that is sometimes the case. And it can also be the case for some people, and other people it's not that. Maybe it's just that the virus is just not very good in this particular case at infecting your cells. And so your body doesn't need to work as hard to get rid of it. And sometimes maybe it's a combination of the two. It's really hard to say.
And like talking about the symptoms, it's kind of difficult. You were mentioning with the grid with all the different types of symptoms you could get. It's really difficult to establish like causality. Like, oh, I have a little bit of a headache and the cough, but the cough could be caused by COVID and the headache could be unrelated, like you're dehydrated.
Yeah, exactly. So exactly why you should take it with a grain of salt.
For sure.
I mean, a lot of this stuff I've talked about, like, take it with a grain of salt, because research into viruses and research into any biological anything takes like years, and then takes months of peer review to be able to make sure that the research you've done is actually accurate and is, stands up to testing. So the fact that a lot of this stuff is coming out just within weeks, that doesn't mean it's not good. It just means that it hasn't gone through all of that rigorous, like, reviewing and everything. So, you do have to kind of...
Like 50% confidence kind of thing.
Yeah, I don't even know what percent confidence to say, but just take it with a grain of salt. Yeah.
Another question I had was when you were talking about macrophages. I know that from another video that was produced about COVID, that it can infect certain cells of your immune system and cause them to overreact and produce too many cytokines.
What other did you find that does that? Sorry.
Another video that talked about it. I think it was posted on the group.
Was it in a nutshell?
Yeah, it might have been that.
Like Kyrgyzstat, or what are they called?
It was like the image was blue, kind of like.
Was it like cute little cartoony like stuff? Yes. I'm glad you mentioned that, because so I actually went and looked at the sources for that video, and I actually quite a few of my sources for my presentation I took from the sources for that video. When it came to the part of it where they said SARS-CoV-2 will infect immune cells and cause them to overreact, I looked at that. They quoted that in their references, and then they were like, we got this because of this. I read in it, nowhere in the research does it show that that is the case.
Yeah, because then they would be tying that into like HIV and like, oh, you have less immune cells, therefore they can't be infected. And
the overall thing that they were saying was correct, is that people get serious illness because your immune system overreacts and because of these cytokine storms. But it's not, there's no evidence to show that it's because the virus is directly infecting certain immune cells. I've seen no evidence for it. They didn't even, and again, in the sources they showed, none of those sources mentioned it. So I don't know why they said it like that.
That's pretty reassuring.
Okay.
And I was wondering actually if it was possible for a macrophage to get infected with a virus after phagocytosing something, or is
Yes, it is. There are some organisms that are able to evade phagocytosis. I can't think of them off the top of my head, but I do remember learning about them.
So they get eaten, but then, haha, that was my plan all along.
Yeah, before they get brought to the lysosome to be digested, they escape
the vacuole.
Exactly.
Fuck them.
Yeah.
Oh yeah, you're talking about the intestine.
Mm-hmm.
So viruses are like this virus is known to bind to the protein, the cellular membrane protein ACE2.
Mm-hmm.
And you were saying in the intestine it was the same protein.
It is.
So have there been reports of the intestines being damaged by COVID yet?
Oh, interesting. So no, I don't think so, except for in the severe cases that I mentioned where like they showed multi-organ damage. In those cases, yeah, I think in some of them they show
that could be sepsis though.
Yeah, exactly. So but in terms of like, have they been like the same kind of huge reaction that we saw in the lungs? I haven't seen any.
Maybe the lungs are affected way faster.
They do. The alveolar cells do express more of those proteins. And also the environment of the lungs, it's open space, and then the cell, whereas the intestine, it's liquid. The small intestine, you're in this low pH liquid. It's a very different environment. I do know that a lot of viruses that do have envelopes don't tend to survive well in the gastrointestinal system because usually the acids break down the envelope.
And that's the good leeway to my next question about the different surfaces. Why is there a different survivability on, let's say, steel and plastic? Is it the resources that the virus can access, or
no? Because the virus isn't accessing anything. The virus can't do shit.
Alive, so.
Yeah, viruses aren't alive. They can't do shit. They just float around until whoop, here's a receptor to something I can. Ooh, and suddenly it is moving around, and but like even when I'm mentioning, it's always our cells and our cellular proteins that do the reactions that, like, when the initial cut, the furin that cuts the binding, the spike protein, opens it up. The reason they need furin to do that is because they can't do it. They don't. Their enzymes don't work yet until they're inside a cell using the cell's energy.
So when you're looking at a virus on a surface, the only thing you're going to be looking at is the phospholipid bilayer.
And the proteins that are in it as well. And so that kind of goes into what Brendan asked. So like maybe why can it survive longer on certain surfaces? I don't know enough about chemistry to be able to answer that, but it's likely that the interactions with the membrane proteins and the membrane itself it's maybe just more stable on certain surfaces rather than others.
Also,
plastic is very inert, right? So it's stainless steel, very inert. So it makes sense.
And why cardboard? That's so strange.
I don't know.
I think they tested cardboard because people are getting deliveries and packages and things.
It makes sense why they tested it, but why it survives that certain amount of time as opposed to others, I don't know. But cardboard is carbon based, right? So maybe that has something to do with it. Maybe some chemical reactions occur over time. Remember that enzymes are just things that facilitate chemical reactions. They don't technically create the reaction. They just make it much more likely that it occurs. With enough time and with enough energy or heat or whatever, things start to react on their own sometimes. Usually not the way you want.
Last question, and then I'm going to let you go.
Thanks.
Sorry.
It's okay. I chose to do this.
Actually, it's not a question. It's just like maybe an add-on to like with the washing of hands, like temperatures that are above like 25 degrees, preferably, to like degrade like the phospholipid bilayer, and a lot of rubbing.
Yeah.
Which would actually like
Yeah, washing your hands properly. Thankfully, my roommates, I did a presentation for them before on pinworms, so they all know about proper hand washing because, yeah, that's important.
Well, thank you very much for this awesome presentation. It was really interesting.
My pleasure. I apologize for how long it is. We have some other questions from viewers.
Hi, Patrick.
Hi, Ben.
Okay, so we got a couple other questions here that we wanted to ask. The first one is, does animal fur transfer the virus? Should dogs also be practicing physical distancing?
The answer is actually yes. They do recommend in the CDC and the WHO that pets should practice, if you're sick, that you shouldn't be rubbing your face into your pet, because the viral particle can survive on the fur for an amount of time. They haven't tested how long, but long enough that if you live with someone else, they can go and it can infect them.
Very much just like other surfaces.
Exactly.
Also, physical heat environment, things like we were talking about the flu being possibly, coming up more often during the colder months. Mm-hmm. Since we haven't had the experience with this virus yet into the hotter months, we don't really know for sure, but I have heard anecdotally that warmer climates do have an effect on its survivability, let's say, or infectivity rates.
So there are so it really depends on the pathogen. There are some pathogens that they thrive in warm environments. That's why you see a lot of outbreaks in parts like in sub-Saharan Africa or in South America and Central America, places where it's humid and it's hot. A lot of places like that, a lot of viruses do like that, but some of them don't.
Do we have any idea about COVID-19?
I don't think so. Not that I've read. The last time I read about it, they weren't sure. I'm trying to think of just like regular SARS, like the other SARS. I don't think it had much seasonal variation, so it's possible. That's also another thing, and some of them don't seem to really care one way or another. So don't know.
Another rumor I heard was about alcohol and consumption, whether that affects the survivability of the virus in the human body. What do you think about that?
Interesting. Probably not. Because the virus is, first and foremost, is in your lungs. It's not actually going to be in your bloodstream, for the most part. And therefore, alcohol isn't going to be destroying it. If you have alcohol to the level where it's actually destroying the virus that's inside your lungs, then I think you have other problems.
Good to know.
Yeah. Yeah, don't do that.
Well, you did mention that, the virus, because it can attach itself to other places in your body, like in the small intestine, perhaps, that's affecting other parts of the body, and it's not just respiratory, or is it specifically talking about because I've heard it's a respiratory virus, but just anecdotally, I know a lot of people, apart from the very severe symptoms, have not experienced necessarily the respiratory, let's say, aspect of the virus.
Yeah, and I agree. And, our roommate, he was very much like that. And so it might be the case that there are more cases of people who are experiencing COVID-19 as a gastrointestinal infection but we really don't know much about it because all we're hearing about is the lung infection. It does appear that, if it is the case that it seems that if it's gastrointestinal, it doesn't last very long, which, like I said, makes sense because it's an envelope virus. That low pH environment, it's not going to do well in there. But so it makes sense that it would do better in the lungs as well. But yeah, that's an interesting thing to think about.
Just to kind of maybe go right back to the very beginning, we've heard about this virus, SARS-CoV-2, coronavirus, COVID-19. Maybe you could just kind of explain
The difference.
why we're hearing why do we have 3 different names for this thing?
Yeah, that's actually a really good question, and I actually meant to explain better like what the differences are. Ben asks like, what is the difference between coronavirus, COVID-19, SARS-CoV, all that stuff? So basically, this is how it works: is we have the virus itself that is causing this pandemic is called severe acute respiratory syndrome coronavirus 2. And a lot of people have just shortened that to coronavirus. The reason some people use coronavirus or say the reason I'm saying SARS-CoV-2 is because there are different coronaviruses out there. Like the original SARS was a coronavirus and its official abbreviated name is SARS-CoV. So SARS-CoV-2 is just another abbreviation for it. For what coronavirus disease 2019 is that's the disease that's caused by SARS-CoV-2. So SARS-CoV-2 is the virus. Coronavirus disease 2019, or COVID-19, it's just an abbreviation, is the disease that is caused by SARS-CoV-2 or is caused by the coronavirus. Essentially, a lot of the time they're used interchangeably. So if you tell someone I caught coronavirus, they're going to know that you don't mean the common cold, right? Because you wouldn't say coronavirus, you just say I caught the cold. You could say COVID-19, even though that technically means the disease. That's okay because you did catch the disease if you caught the virus, so that makes sense as well. I was just kind of trying to be more technical with SARS-CoV-2.
I mean, SARS-2, the sequel, sounds way catchier. Once
SARSing.
Once you explained what
right?
What it was, right?
Miss Rona if you're nasty.
So I think that's interesting too, because I would say in the common parlance of most people, I don't think they recognize I don't think a lot of people knew what SARS was in the first place.
Yeah.
They just knew it was SARS, or H1N1 was the swine flu. They've heard these buzzwords, but they didn't really understand what it was trying to be an abbreviation for.
What's interesting is that there have been a lot of seasonal flus that have been H1N1 flus, but they're still different from the H1N1 pandemic flu that came out in 2009. So a lot of our naming conventions are not perfect and are overly complicated. And that's just kind of also the nature of viruses themselves, that because they mutate so much and because a lot of the times they're not new species, they're just different strains and stuff like that with only a few mutations, it can get real hard to track.
And you told me to, this coronavirus or COVID as an abbreviation. Why is it COVID?
So co as in corona and vid is virus. And then I don't know where. Yeah, virus. So VI and then D because disease. So COVID-19.
And it's 19 because it was first
discovered in 2019. That's when the initial infection is believed has occurred. And that's when China first announced, like, hey, yo, we got something up here.
Not because it's the 19th one.
Not because it's the 19th one, no. It's because of the year. Yeah.
All kinds of great information here. I do have one final silly question. Would you rather fight one horse-sized Viron or 1 billion Viron-sized horses?
Oh my god, that is a really good question. I'd fight one. I'd fight one horse-sized Virion because Virions still are Virions, which means they can't do shit. They're just gonna fucking sit there. I can just blow it up and won't do shit. Whereas just a ton of tiny horses, they're still horses, they're just really small. That could fuck you up.
Horses are bitey and kicky.
Yeah. I don't want that. Just a big old virus, that's just in the way.
All right. Well, I think that was all of our questions. So thank you for spending your evening with us and answering all of these. Just giving us this great information. I feel like everyone's a lot more informed about what the virus is, the science, and I think actually going back to some of those graphs, we saw those big red curves and the flattening of the curves, and then what might happen if we stop doing things. I think the most misleading thing about those graphs actually was the first one, the red one with the big lump that actually went back down. Well, why did it go back down? Is it was because everybody died? Yeah, Patrick nods his head.
Well, let's look at that one specifically.
This one, right?
Oh, okay.
The collective response, strong collective response, and the short-term only, you see it comes back up. But the first image, why does the curve just go up and flatten out? Why is it not all the way across red and even just growing?
No, you're correct. It's because everyone's dead. I mean, that's the unfortunate. It's either everyone's dead or everyone that's caught it at this point now is like in the hospital and like unfortunately dying. Because when you're dead, you're much less likely to pass it on because you're not coughing.
So
keep at it.
Keep at it. Do your best, and let's keep that curve flat and follow our recommendations from our healthcare providers. Maintain your social distancing, and even though you might not be able to get it reinfected, we don't fully have all of the answers yet, so we don't really know what to do. So day by day, case by case, we're gonna keep on it, keep following our good instructions. And if we have to maintain our distance for a while longer, so be it. We'll all learn how to play online games.
Exactly. It sucks, but, humans are adaptable creatures. We will continue on. And like I said, it won't be forever. Eventually, it will end one way or another. Hopefully, the good way. All right. Thanks to you guys. Bye.